Hi, everyone. Thank you for joining the webinar titled Preparing Vaccine Studies for a Regulatory Submission. I'll start with a quick introduction. My name is Michael Beers. I'm a principal consultant at Pinnacle 21. I work with our clients in helping them use our software and interpret their validation findings. I also help manage the validation rules here at Pinnacle 21. I also work as a data fitness SME for various projects at the FDA, including Jumpstart, Core Data Fitness, and others. If you have a question at any point during the presentation, please use the chat icon on the left side of the application window. I'll answer as many questions as possible at the end of the presentation. For questions that I'm unable to get to during the webinar, we'll try to provide answers on the Q&A section of our website where we'll put the slides. Okay, so for the agenda, I'm going to talk about I'm going to do a quick introduction, then go over the relevant industry guidance for vaccine trials. And discuss some of the common data to vaccine trials, how the data should be mapped, and how the industry is currently handling it. Then I'll discuss common challenges, issues, and then I'll wrap it up. Just a quick disclaimer as well. This webinar discusses some of the key guidance for preparing your STTM and ATOM data for submission to the regulatory agencies. It's not an exhaustive list of guidance for vaccine trials in general, such as the design of trials, conduct, stuff like that. I do not speak in any way for the regulatory agencies or standard development organizations. And as always, you want to consult with your review division. Make sure that they're on board with what you plan to do. Okay, so the goal for all of us in the industry really is to reduce the time it takes to get important medicinal products to the market. It's always important to do all you can to reduce the processing and review times, but during times of public health emergencies, it's even more critical to provide the data as expected to try to cut down any possible time it takes to process and review this data. In May of last year, the government launched something called Operation Warp Speed to accelerate making treatments and vaccines for COVID-19 available as soon as possible. This was successful. A number of COVID-19 vaccines were made available to the public in record time through emergency use authorization. I'm mentioning this because when preparing data for submission, we want to make sure that we always do our part in this process. Make sure that we aren't introducing any potential delays in the time it takes to review these applications. One of the most important ways we can do this is through data standardization. The key to reducing processing and review time is really automation. Any manual processing and review of data is inefficient, time-consuming. Evaluating data quality using any kind of manual effort is very problematic as well. To be able to automate processing and review, the data needs to adhere to standards and guidance as strictly as possible. The same data needs to be submitted the same way across the industry. It needs to be free of any kind of major data quality problems as well. The challenge here is being aware of all the different guidance available and how you should use and implement it, and what to do when they contradict each other, which they usually do. The first type of guidance you need to be aware of is regulatory agency guidance. So for the FDA, we have, as usual, you should be aware of the technical conformance guide. This is where the FDA provides the technical recommendations for how to submit study data. The current version is version 4.7, which was released in March of this year. There's also the Vaccines Technical Specifications Guidance, which was released by CBER's Office of Vaccines Research and Review. And then there's the Conduct of Clinical Trials of Medical Products during COVID-19 Public Health Emergency. This goes into a little bit more of conduct of trials and how to handle unique situations, but this is something that you should be aware of as well. For PMDA, they do have a website, PMDA's efforts to combat COVID-19, and here they have a number of different documents. So if you have a PMDA submission, you want to be aware of this website and all of the documents and guidance that is posted on this website. Two relating to vaccines are the two I have listed here, the principles for the evaluation of vaccines against COVID-19. And then there's an appendix 1, which is for the variants. Again, these go into a little bit more of the study design and contact and not exactly preparing the data, but if you have a PMDA submission, you absolutely want to be aware of these documents and all of them. The other type of guidance that we have is CEDISC guidance. So the first that you should be aware of course, is the implementation guides for the foundational standards, STTM and ATOM. But then there are extensions to these foundational standards called therapeutic area user guides. There are a number of them that apply to vaccine data. The first one, guidance for ongoing studies disrupted by COVID-19. This really just applies to all types of trials, not really vaccine specific, but it goes into how to handle situations in your study due to COVID-19, such as missed visits, deviations, stuff like that. There's the interim user guide for COVID-19. Just to note that the final version should be released soon. At the moment, we have the interim version to reference. There's a section 13 in there that discusses vaccines. And then we have a number of different actual TOGs. So we have the vaccine TOG, which is kind of the overarching therapeutic area user guide for vaccine data. Then we have kind of the indication specific ones, influenza, tuberculosis, Ebola, HIV, and virology. And so each of these kind of touch on vaccines as well. And then this slide just shows you that even looking at FDA guidance, we can see that it's very clear that you need to be aware of the different regulatory guidance, CEDISC guidance. You need to use them in conjunction with each other. Like this first passage I have posted here for the vaccines therapeutic area user guide. This is from the FDA's technical conformance guide. It says that you should use the vaccine TOG in addition with CBER's OVRR technical guidance and also the interim user guide for COVID-19. So you need to be aware of what's in each of these guidances and use them together. The second passage, again, is from the TCG, it's Appendix D, and it's saying that you need to be aware of certain guidance in that guidance for ongoing studies disrupted by COVID-19 pandemic. Like they go into how to handle the subject visits domain. In that they're specifically requesting that some new variables be included as parent variables in the SV domain and not be included as a supplemental, the sub-SV domain. And just to note that I believe this request from the FDA, it will be updated in CEDISC SDTM IG 3.4 whenever that's released. So likely CEDISC will update their guidance to reflect FDA requests here. So as for the data common vaccine trials, well, there's a lot of data that's common to all clinical trials. These that I have listed here are typical types of data that you would find in a vaccine trial. The first four are mostly routine types of data. So we have the tests for the data, or tests for the disease being vaccinated against. We have the details of that disease being vaccinated against, vaccine administration, and antibody data. Those are mostly routine mappings. There's a spot for these types of data in the CEDISC standards. You can refer to the tags, and I encourage that to see. There are some specific things that you can do, additional variables that you might need to handle some of this stuff, but again, it's mostly routine mapping. The next three are what I'm going to focus on in this webinar. There's risk factors for the disease being vaccinated against. There's efficacy endpoints. Those two I'm just going to touch on briefly. And then I'm going to spend most of the time on reactogenicity data. Okay, so for risk factors for the disease being vaccinated against, if we look at the CDISC therapeutic area user guides, you can see the COVID-19 guide shows that pre-existing medical conditions mapped to MH, smoking mapped to SU. So these are common types of data, and there's a spot for them. Doesn't really depend on new vaccine guidance. But then there are additional information like PPE, travel, contacts, exposure to animals. These are all mapped to a new draft domain, environmental and social factors. If you look at the vaccines and influenza tags, there's nothing really specific to that. The Ebola and tuberculosis tags also map some of this data to that new ER domain, which is an events domain. You can see at the bottom I posted, this is a draft domain. It's a draft events domain, but this was introduced to handle this type of risk factor data. The CBER OVRR guidance doesn't really discuss this. So again, we go back to the CDISC tags for guidance. And then if we look at how the industry is currently handling it, this data is generally mapped to the subject characteristics domain or the sub-DM domain. It seems that there's some hesitancy to use this new custom domain at the moment. Not sure if that's just because this data had typically been mapped to these two domains, SC or sub-DM, so sponsors or CROs keep mapping it to that same stuff, or if there is just hesitancy to move it to a new custom domain. If there is, recommend discussing this with your review division that you plan to use this new domain modeled in the CDISC therapeutic area user guides, or you could email the eData email address. But again, the recommendation is to start using this domain. For efficacy endpoints, if we look at FDA guidance, we see that in the CBER OVRR guidance, we see that efficacy data, when related to clinical disease, when that's the objective, then this data would typically go to the CE domain with supporting information going to domains like MB, FA, CE, VS, and PE. They provide a way to differentiate this data. They have a CE cat value. You should use the word efficacy as your CE cat. Test results confirming this disease should be mapped to, in general, mapped to MB. But then they request a new flag to be added to the sub-DM domain. So this would be the clinical disease endpoint case flag. If we look at CDISC guidance, this is something that's not covered. They do state that they do plan to possibly cover efficacy information in a subsequent version of this guide, but at the moment there is nothing. And if we look at how the industry is currently handling it, we do see that sponsors or CROs are starting to adopt this clinical disease endpoint case flag, but it's not widespread yet. And this could be because you would need to be aware of that specific FDA guidance. And again, it's not listed anywhere in CDISC guidance. So if this is then modeled in a subsequent version of a TALG, we would hope that preparers would be more aware of this and start to implement and give the FDA what they request. Okay, so moving on to reactogenicity data. So this data is the typical and expected adverse reactions, related to an administration of a vaccine. These would either be local or systemic, and they're typically solicited for a certain period of time. Typically it's 7 days after the administration of a vaccine. But I guess it doesn't have to be 7 days, but it's a typical assessment period. So some of the common reactions are, for systemic, you'd have fever, myalgia, fatigue, headaches, nausea, chills. Administration site reactions would be something like redness, swelling, pain, tenderness, stuff like that. But this data is unusual because the mapping is complicated. So typically, data that gets collected for a clinical trial gets mapped to a single domain. There are exceptions, but that's a typical way that it's handled. For this type of data, that's not the case. You have your daily assessments collected. They go into a findings domain. You have then global event records created in another domain, the CE domain, and then you might even have an adverse event record created depending on that data. So it is kind of a complicated set of data to work with. So if we look at CDISC, vaccines, TAUG, and how they modeled this data, they do provide 3 different ways to handle it. The first method is the flat model. This is where you have records from your daily assessments, usually collected using an e-diary. They get mapped to the FACE domain. So for that assessment period, the one I have listed, shown here is a 3-day assessment period for simplicity, so they would fit on the slide. So for each of those 3 days, they assess a number of different reactions and whether they occurred or not. If they did occur, then they might collect more information. So if we look at this example, it's the middle set of data here. This is our FACE domain. We have 2 sets. I have the green rows are for headache, and then the blue rows are for erythema. So you can see for headache, there's 3 assessments, whether it occurred or not. It did occur twice. When it occurred, we also collect the severity. So one was a moderate headache, one was a mild headache. Then these headache assessments all get rolled up into a global record in CE. So you can see the top record highlighted green is a headache record. It's a systemic reaction. CE occur is yes. You can see that the severity is moderate. That's because for that assessment period, we take the most severe value of severity and use that as our CE sev. You can see that the CE start date matches the first occurrence in FA. So the FADTC value for the first occurrence, and then CE end date would be the FADTC value for the last occurrence of headache. For the blue records, we can see erythema. It was assessed 3 times. Each time was no. So the value of FAORES is no. They again get rolled up into one global erythema record, and you can see that CE occurs no. Start date, end date are both blank because it did not occur. And then the bottom one is a set of VS data for temperature records. We can see these get, again, rolled up into one fever record in CE. Again, CE occur is yes. CE start date is the first occurrence, VSDTC for this fever. And then CE end date is the last occurrence of fever from the VS domain. The next way to model this data that is listed in the TAUG is the nested model. Here we have the same data, but you can see that now for my FA domain, I am no longer mapping that set of erythema records. Since it was assessed during the assessment period and each one was no, these records are it's not necessary to map these to the FA CE domain if you're using the nested model. It still does roll up into one erythema CE global event record where the CE occur value is no. So this is really just kind of to, I suppose, save room in your FA domain to make that data set not too large. And then if we go to the third model, this is the highly nested model. This is even more complicated. So now you have, again, that FA domain erythema was assessed as no for each each day, so that is dropped. That is not mapped to the FA domain. Instead of rolling up into one erythema record, instead you're introducing global category records into CE. So you have a systemic event record. Occur indicates if there was any systemic event that occurred during that assessment period. In this case, there are two. There's headache and fever. So you can see it again. They correspond back to the FA-CE domain and the VS domain. And then we have one record at the bottom here that's introduced for administration site event. Since there are no occurrences of erythema or anything else in my example here, the CE occurs no, and that's it. We don't add any additional global event records to it. So 3 different ways to model the same data provided in the talk. So then if we look at what the FDA guidance says, the CBER OVRR vaccines technical specifications guidance says that they prefer the vaccine TAUG, the flat model, to be utilized. So a clear direction from the FDA, that's the model that they prefer. They do make an exception. The nested model could possibly be necessary if your trial is large enough and you have a significant amount of data. In that case, you want to contact the review division and see what they say. And if we look at how the industry is currently handling it, in most cases, the flat model is utilized. That's where we have each of those daily assessments collected in the eDiary, mapped to the FA-CE domain, and then get rolled up into global event CE records. Okay, so the first challenge, reactogenicity events that last longer than that assessment period. So typically, or commonly, it's a 7-day assessment period. So they assess a certain reaction each of those 7 days. If a reaction occurs and it lasts longer than that 7-day or whatever assessment period, the FDA guidance specifies that that would then be considered an adverse event and should be mapped to the AE domain. You can see I have a passage listed here with highlighted yellow that is showing that the FDA does request that get mapped to the AE domain. However, if we look down at the next passage, this is from the CDISC vaccines TAUG. There's an example where they have a reaction that lasted beyond that assessment period that was not mapped to the adverse events domain because it's not a protocol defined adverse event. I think this is misleading because then this is saying sort of that you should rely on whatever your protocol defines and ignore FDA guidance, which I think is a bad idea. So I think you definitely want to follow what the FDA requests, or at least discuss with them if you plan to go against that. There is another example 3, that does show in that vaccines TAUG from CEDIS that a reaction continuing beyond the assessment period is mapped to the AE domain. However, they don't show how to map the duration variable in each domain, which is another complication that I'll get to in just a moment. Okay, so when you do this, when you map a solicited reaction that occurs lasts beyond the assessment period to the adverse events domain, you want to make sure that the adverse event start date and end date matches exactly what the clinical event start date and end date are. Like I said, then there's a further complication of how to map the duration. So FDA guidance says that you should treat the duration during the assessment period separately than the duration of the continuance period. Therefore, when you look at these dates, when you look at the period of adverse event start date to adverse event end date, that's not going to match up to your AE duration exactly in most cases. And here's an example of how that would look. So here we have a 7-day assessment period for fatigue. So each day fatigue occurs, and then each day they collect another record for severity. So it's moderate every single day. And we can see that that gets rolled up into one global clinical event record for fatigue, where the start date, the CE start date, matches the first occurrence of fatigue from the FACE domain. The CE end date, you can see, is 2 days after the last fatigue record assessment in FA. So this means that fatigue continued beyond that assessment period. You can see below that now we have generated an adverse event record with the same exact dates as the clinical events start and end date. However, the durations differ between these two domains. So for CE, the duration is 7 days because the reaction lasted for those 7 days during the assessment period. For the adverse event, it extended 2 days past that. So that's why we have the 2-day adverse event duration. So other considerations when you do this, when you map solicited reaction to the adverse event domain, you want to use, for AE term, you want to use the exact same CE term. There's no real guidance stating what to do, but it would just make sense so that it's easy to link these two domains together and determine that you're doing this correctly. So the recommendation is to use CE term in the exact value in CE term as your AE term. And then how would you differentiate these from your other adverse events in your adverse events domain? CBER OVRR guidance does provide a category that you should use. You should use the word reactogenicity. And then the other thing is that these, in analysis, these adverse events are usually removed from the ADAE domain and instead mapped to a different atom data set, which is a little bit odd to move these reactions into the adverse event SDTM domain only to separate them back out in analysis. It seems a little bit unusual to be required to do that. When you do that, this results in an ADAE versus adverse event traceability validation rule, and then you're required to explain that. So if this is the case and you do in analysis, you move these reactions out of your domain into a different atom data set, then you'd want to explain it appropriately. You'd want to list exactly what you've done as your explanation. Okay, the next consideration for mapping this reactogenicity data is the topic of CE start date and CE end date. The COVID-19 TAUG says that it lists a passage from the FDA's TCG saying that if an event occurred, you should also collect the start and end date, and that's what gets mapped as your CE start date and CE end date. The CDISC vaccines TAUG also goes into how if only daily diary collection dates were collected, then you would leave CE start date and CE end date blank. However, this is not really how the industry is handling it, and I'm not sure this is how the industry should handle it. The way that the industry handles it now is to take the minimum FADTC for that reaction as your clinical event start date, and then the maximum FADTC for that reaction for that assessment period as the end date. So that's not every sponsor is doing that. A lot of sponsors do have a way that, if an assessment occurred, then it does trigger the new fields that would collect the start and end dates, and then those would get mapped to CE start date. But if you don't have that, seems like a better idea than leaving those start and end dates blank would be to use the minimum FA DTC and maximum FA DTC. So it would be a good thing for this to be discussed in more detail in a subsequent version of the CDISC vaccines TALG or even FDA guidance to kind of clarify how this data should be mapped. Another consideration is that you'll be mixing reactogenicity events with other clinical events in your CE domain. And so how do you differentiate them? CBER OVRR guidance, it does list some values that you should use, so we definitely recommend using these values. For reactogenicity events, you should use that reactogenicity term, and then you should further subcategorize it with administration site or systemic. And then for your efficacy data that gets mapped to the CE domain, you want to use your category as the word efficacy. And so there is no CDISC control terminology for these category variables in CE or even AE. So you're kind of required to, pick out these values requested by the agency in this guidance, which is a little bit less than optimal. So hopefully at some point CDISC would provide some kind of control terminology and kind of force sponsors to use these exact terms, just so that sponsors don't think that they're able to use whatever they want since they're not subject to control terminology. The other issue is reactogenicity events not measured on a certain day. So if you have a mix of data in CE that's not pre-specified, and then you mix in your reactogenicity data and you leave occurrence no because it wasn't measured on that day, then you could run into some problems not knowing what a null value of CE occur means. So in this case, we do recommend definitely using the CE stat variable to indicate that this was not done, and then use the reason not done to describe why this assessment was not done. Another consideration is controlled terminology. So there is some controlled terminology, CDISC controlled terminology for some of this reactogenicity data, but not all. We do have FA test codes and FA tests that are control terminology that's available, and here's that list. So we have COVID-19, Ebola, tuberculosis, and vaccines, findings about test codes and tests. So you should absolutely be aware of this and use those. But the problem is that there is no control terminology for FA-CAT, FA-SCAT, CE-CAT, AE-CAT, stuff like this. So sponsors could interpret that as they can use whatever they wish. However, we do know that values are specified in that FDA guidance. So until CDISC guidance is updated to require control terminology for these variables, and these values are added to the CDISC CT, right now sponsors and preparers of the data would need to be aware that they need to search for certain guidance, like FDA guidance or even a TALG, pull out these values and use that. Otherwise, if sponsors are to use their own values, then it's really not standardized data, and it makes processing, review, validation, all of that very difficult. Some other issues, sponsors might use FA instead of FACE. They might, keep it all in one FA domain, or if they only have this type of data, this reactogenicity data, they might think that they don't need to split to use FACE and just put it in FA, but then that does make it a little bit challenging where some studies are submitted using FACE and some are using FA. It's a minor challenge, but it could be one. So I guess my personal recommendation would be to just always use FACE for a vaccine trial. It really just indicates that this is your reactogenicity data. And then other times sponsors use variables like epoch to indicate vaccination 1, vaccination 2, even though all the guidance kind of shows examples and goes into using the TPT ref variable. And so while it could potentially be argued that values of vaccination 1, vaccination 2 do fit into the definition of the epoch variable, we'd still recommend, strongly recommend using this TPT ref. Again, we want all data, all similar data to be submitted the exact same way across the industry. If one company uses one variable to indicate this and another company uses a different one, then it makes processing a lot more difficult and manual adjustment necessary to handle that. Another issue is that there's no guidance for how to map analysis data for your vaccine trials. So I listed out each relevant guidance here. It's either that there's nothing related to ATOM in these therapeutic area user guides or FDA technical specification guides, or that the TAUGs say that future iterations or subsequent versions may include updates for how to map this ATOM data. And so hopefully this does happen, and we do get updated guidance, which kind of goes into how this data should be mapped in ATOM, because the problem is, if you don't have any guidance, this is the kind of situation that you result in. Here we have 3 applications from 3 different sponsors. These are just samples. Where application 1, you can see that there are 2 ATOM data sets related to your reactogenicity data. There's one for events, ADCE. There's one for your reactogenicity findings analysis, which is named AD-REACT. They're both BDS data sets. Application 2 has only one reactogenicity related ATOM data set, and it's named ADSR, so it's a completely different data set name, and it's also a different structure. It uses the occurrence data structure for this. And then application 3, we see that, again, there's 2 different reactogenicity data. One is AD-FACE, which is a BDS, and then there's an ADCE, which is a occurrence data structure. So similar type of data, reactogenicity data, mapped in completely different ways across 3 different sponsors, 3 different applications. So that's what makes guidance so critical, that a lack of guidance results in situations like this. And as you can see, this is not standardized data. So hopefully guidance will be updated to provide mapping for this. Okay, then just to wrap it up. So what should sponsors and CROs do? You really do need to be aware of all the different guidance available for mapping each type of data, and it is very confusing. Like I showed, you need to be aware of your implementation guide, the different therapeutic area user guides. Not only that, those therapeutic area user guides could reference a different therapeutic area user guide, or you might have a specific type of data that you would then need to dig through and find in a different TAUG. So it does get very complicated. And then there's also the FDA guidance that you should, if anything, you should prioritize, because again, this is the authority that will be reviewing and approving your application. So you don't want to contradict, or you don't want to go against anything that they say. So when you do have this contradiction, you probably should prioritize the FDA guidance, or if you have concerns, you would contact them and ask them how to handle it. What should CEDISC do? Hopefully at some point update guidance to remove maybe that highly nested model. That could just be confusing if the FDA lists that they prefer the flat model, and with exception possibly the nested model. It seems just unnecessary to provide a third way that agency doesn't even mention. So again, removing that would probably be a good idea. Also creating controlled terminology for reactogenicity data, so that preparers don't have to like scour through the different TAUGs and guidance to find values and pick them out and then plug them into your data. It would be good to incorporate other pieces of FDA guidance into the new versions of the TAUG, and it seems like CEDISC does do that, but it's good to always keep updating them and to align with FDA guidance. You want to avoid all the conflicts that you possibly can, because that's where you get different sponsors doing different things because of these conflicts. And then CDISC should also create guidance for analysis data and include these in the various tags. And what should Pinnacle 21 do? So without enforcing the use of this guidance, then the widespread implementation is less likely. It does occur because sponsors are good at following guidance and updating their standards and their mapping, but the way to really enforce it and get everybody on board and to use this and to get actual standardized data is enforcement, and that would be done through checks. So our job is to create validation rules for this type of data. There are some challenges, but we are investigating this at the moment, and we do hope to introduce some new validation rules specific to reactogenicity data and other types of vaccine-related data. Okay, I believe I have just a few minutes for some questions, so I will start to answer some of these. Okay, so the first question, will the slides be available after the webinar? Yes, we'll definitely post the slides. We'll post the webinar, the slides, and any other associated information related to it after the webinar, and this will go onto the website, and you should receive an email notification with a link when that gets posted. We provide links to the guidance. Yes, so in my slides, I do provide links to some of the guidance, and again, the slides will be posted to our website after the webinar. Okay, so what should I do if my vaccine study is not covered by a CEDISC tag? Well, if your vaccine study is for an indication that doesn't have an associated tag, you'd still definitely want to follow the guidance I discussed in this webinar, and that would be the FDA guidance, like the CBER OVRR guidance for submitting vaccine data, and also the vaccine tag released by CEDISC. So there definitely is guidance that you should be aware of in that case. Next question, should I follow CEDISC or FDA in case of conflicting guidance? My recommendation is to always give the regulatory agency what they requested, or at least discuss your concerns with them and ask them how they should do it, how they recommend that you should prepare your data. Or you could even email eData. That would be an option as well. Another question, does Pinnacle 21 provide consultative services for vaccine submissions? We have provided that service actually for clients previously. So if that is something that you're interested, please do reach out to us and we could always discuss the possibility of that service. Another question, will Pinnacle 21 support TAUG compliance? Yes, we plan to, as much as we can. There are some challenges. I actually wrote a paper related to this, to the challenges of implementing and enforcing implementation of the TAUGs with validation rules. So you may be interested in reading that paper. It's titled The Need for Therapeutic Area User Guide Implementation. I presented that at both FUSE and Pharmasoc, I believe, in 2019. The next question, how do you link the data between all domains for reactogenicity data? That's a good question. So the tags show that this data is linked between the different domains using the link group variable. So we recommend using that. That's a variable that fits for that purpose. Okay, I believe that I am out of time. So I wanted to thank everyone for joining and have a good day.